Hey everyone, this is your lesson on genetic drift, one of the forces of evolution we are going to be talking about. So what is genetic drift? Genetic drift is a change in the frequency of an allele due to random events or chance. It's very important that you understand that this is due to randomness. It's going to affect smaller populations more than it affects larger populations. And it can be broken down into types of genetic drift. Now, when we talk about random events, these random events can be solely through the formation of the gametes. So in independent assortment, how the chromosomes will line up randomly on either side of metaphase will affect or is known as genetic drift. There's also randomness in the fertilization and in general the production of the offspring that's going to survive in the next generation and represent the next generation. So that randomization can be due to just the formation of those gametes. Other types of genetic drift are known as bottleneck effect, which is usually by a disaster, where a disaster wipes out part of the population, but this could also be due to selective breeding or overhunting and overfishing. And then the last one that we're going to talk about is what's known as the founder effect. This is when a group of individuals, usually smaller in number than the original population, move to a new area. And in all of these cases, number 1, 2, and 3, what we're looking at is how the frequency of the allele is going to change. Now, in some cases, alleles can become fixed, meaning that due solely to chance, one allele in the next generation can make up 100% of the population, and some alleles may be completely lost in that population. So we're going to be modeling these concepts. So we're going to be doing this assignment called Genetically Drifting Worms, and this can be found in your notebook. And one of the things you're going to notice is that we have several generations multiple times. In fact, I like to go ahead and divide these populations up. So this row right here represents multiple generations of one population. And what we're going to do to illustrate genetic drift is we're going to do the same thing with the next row and the next row and the next row. So the first thing you're going to want to do is you're going to want to designate your alleles in this population. And we're going to do this in that first generation all the way on the left. So what I have done is I have colored in each of the starting worms. There are 5 of them in this population. And I've colored them different colors to represent that they have different alleles. Now, the carrying capacity of this environment is only 5, which means that every generation there will only be 5 worms. To model genetic drift, what we're going to do is randomly select which of the 5 represent the next generation. You can do this by asking a friend to pick a random number 1 through 5. You can ask Siri to pick a number 1 through 5, or you can even just type into Google random number between 1 and 5, and it'll bring results up. Hey Siri, pick a number between 1 and 5.
A random number between 1 and 5 is 3.
So Siri picked the number 3 for me, and what I'm going to do is I'm just going to put a mark next to number 3 so that I know it's passing on one of its gametes to the next generation. And I'm going to repeat this until I have 5 eggs for the next generation. So these are the results of what Siri designated as random numbers for me. You can see here how that she picked 1 once, 2 once, she picked the number 3 twice, and then the number 4 once. Never did she say number 5, so that allele is now lost in the population. So in the next generation, what I'm going to do is I'm going to color it according to the gametes that were produced. So here is the next generation. We had 1 gamete form here for number 1, 1 gamete here for number 2. We had 2 gametes that were blue, so in the next generation there's 2 blues, and then the last one is gamete number 4, which is why we have the green here. This allele here was removed from the population. Then what I'm going to do is I'm going to repeat the same process using these numbers now to figure out what the next generation is going to be. I am once again going to ask my friend Siri to select random numbers between 1 and 5. These are the gametes that resulted from her random guessing, and now I'm going to fill in the next generation. I'm going to repeat this process until I get a fixed allele. In other words, I'm going to repeat this until all worms in this population are identical. So as you can see from my example, it took 5 generations before the blue allele became the fixed allele. What you're going to do for the rest of the sheet is repeat the same process, starting with the same original population here on the left, and again randomizing it to see what the next generation is. Then, when you have the next generation, you're going to again randomly select which of these moves on to the next generation. What we want to see in these 5 populations is even if we start with the same original alleles, do we end up with the same fixed allele every time? Or does it end up being different alleles that are fixed in each of the populations? We also want to see, does it take the same amount of time for each generation before an allele becomes fixed? In the next activity, you are going to be modeling the founder effect. Now, this is not in your notebook, but it is on a separate document linked or assignment linked on Schoology. We have here a population of wild M&Ms, and we're going to treat each color as an allele. Now, to model the founder effect, what we're going to need is we're going to need the original population frequencies of each color. For the assignment that you have on Schoology, you can use either your own candies, or I have uploaded a picture for your convenience as well. So I'm going to calculate the frequencies of each of the colors by counting up the number of each color and dividing it by the total. So the number of each color of M&M divided by the total number of M&Ms in my population is going to be its frequency. So I have 101 total M&Ms on my screen. I'm going to count up now the total number of red and divide it by 101. So I have 29 red, and when I divide that by 101, I get a frequency of 0.29 when I round to 2 decimal places. I'm going to do the same with the rest of the colors. So these are the frequencies of each of the colors in my original population. For the founder effect, what we're going to do is select an area that contains 10 M&Ms, and these 10 M&Ms are going to migrate to another area. They're going to separate from the main population. So the area I've circled in pink are the 10 that are going to migrate to a different area, but I could have easily selected any other 10. Now what I'm going to do is I'm going to go back and recalculate the frequencies of this founder population. Only this time, when I count the number of each, what I'm going to do is then divide by 10, because there's only 10 in the founder population. So these are the new frequencies of the founder population. And what you'll notice is that the frequencies are different from the original population. In some cases, the frequency increased slightly. In some cases, the alleles, or the frequency of the allele, went to 0 because that allele was completely lost from the original population in this new founder population. So depending on which of your M&Ms migrate from the area, the population or the frequencies of the alleles may not be necessarily representative of the original population. This is what's known as the founder effect. Now I want to model bottleneck effect. What happens with the bottleneck effect is that there's usually a disaster that will wipe out a part of your population. Now, it doesn't necessarily have to be exclusively a natural disaster. This could be something like artificial selection, where certain colors are bred because they are wanted in the next generation. This could also be due to something like hunting and removing members of a population. So what I'm going to do, since this is the same background or the same population of M&Ms, I'm going to bring over my original frequencies from the previous slide. So these are the original frequencies. And now, due to some natural disaster, certain areas of this entire population are going to be affected. So I'm going to randomly select this area that got wiped out by a tsunami. And the remainder of the population was far enough inland that they weren't affected. Now I'm going to recalculate all my frequencies of the surviving population. My new total for this population is 39. So here are my new frequencies. You can see that the red is pretty similar to what it was before, as was the orange. But as you notice the yellow, the green, the blue, and the brown, there are some significant changes between this population. Now, this is considered a part of genetic drift because the size of the area that was affected and the organisms or the individuals that were in that affected area is completely random. Now, when we're talking about genetic drift, it's important how this is going to affect populations, especially when we look at conservation. Keep in mind that larger populations are less susceptible to genetic drift than smaller populations. But one of the ways to overcome genetic drift is a process known as gene flow. Gene flow is when another organism from a different population comes in and adds to the gene pool of the future generations of the population. This will help restore lost alleles. So for example, this is in Utah. This is what is known as a wildlife corridor. This area over here is completely separated from this area over here by this giant highway that's in between them. This is essentially stopping organisms from each side coming over and mating or gene flow. So what the state of Utah did was build this wildlife corridor, and this is going to allow organisms to safely cross this area because it's still wild. It still resembles their natural area. They have a tendency to use it more often than crossing here. They're going to cross over, and any alleles that might have been lost in this population, if they're present in this population, can come over, and that is a process known as gene flow. So you have 3 tasks to model this process of genetic drift. You have the genetically drifting worms in your notebook, as well as an activity in Schoology that has 2 tabs. One that represents founder effect and one that represents bottleneck. Thanks for watching, y'all. See you later.